Good morning or good afternoon, everyone, depending on where you are joining us from, and welcome to today's webinar. My name is Kalina from Business Review, and I will be your host today. It is our pleasure to have Medpace with us today, who will be presenting this webinar titled Avoiding Common Pitfalls in Cell and Gene Therapy Trials. Today's guest speakers are Jan Ochocki, PhD, Regulatory Submissions Manager, and Elizabeth Shepard, PhD, Clinical Trial Manager. I would like to welcome you to our webinar platform on 24. You will notice that this webinar is browser-based, so if you disconnect for any reason, please just click on the link that you received via email to rejoin the session. In order to ask any questions, you can send those in via the questions widget. Just type them into the box at the top corner of your screen and click Submit. Towards the end of the session, we will allocate some time to address any thoughts or questions that you may have. Please use the yellow Help widget if you require any assistance, and know that you can also move, resize, and maximize any of the windows in front of you to get a better view of the slides. But now, please allow me to welcome Jan Ochocki. Jan, over to you.
Thank you, Kalina. Good morning, good afternoon, everyone. In this webinar, we will cover the following. We will start with some background information on cell and gene therapies. Then we will discuss the regulatory considerations, followed by operation considerations. And then we will discuss the hurdles for cell therapy trials, and then share a few case studies and best practices. So, advanced therapy medicine products as known in Europe, or cell and gene therapies as known in the US, are innovative group of medicines based on recombinant nucleic acids or engineered cells and tissues, offering groundbreaking new opportunities for the treatment, prevention, and diagnosis of disease. And the novelty and complexity and technical specificity of these therapies pose specific regulatory and operational challenges to the conduct of clinical trials. And in addition to that, regulations and guidelines surrounding these therapies vary greatly between countries, posing regulatory and operational challenges for companies conducting global clinical trials. And our key message throughout this webinar will be that it is crucial to align the overall product development strategy with regulatory, clinical operations, manufacturing, and supply chain requirements to facilitate the clinical development of advanced therapies. And we will show you in this webinar how this can be achieved. So before we go into regulatory considerations, I would like to introduce Medpace and talk a little bit about our experience in advanced therapies. So Medpace is a global full-service CRO founded in 1992. Our ATMP experience is fueled by a team of medical directors who are leaders in the field and the knowledge of our advanced clinical practitioners and clinical trial managers. We have conducted over 70 trials, 70 advanced therapy trials across 28 countries around the world. And our investigational product portfolio includes allogeneic and autologous cell therapies. We have also worked on ex vivo and in vivo gene therapies, and also gene editing technologies. So there are different types of advanced therapies. In the US, we have gene therapy products, human somatic cell therapy products, and combination products. Whereas in European Union, we have 4 different types, and these are gene therapy medicinal products, somatic cell therapy medicinal products, there are tissue engineered medicinal products, and there are combined advanced therapy medicinal products. And for the purpose of this webinar, we will focus on the cell therapies and genetically modified cell therapies. So there is a huge area of regulatory considerations for advanced therapy trials, and these are shown in this slide. And in the next set of slides, we will demonstrate how we address these considerations during the planning phase of clinical trials and startup. So for regulatory teams, the most common pitfalls in advanced therapy trials are the lack of strategic engagement with the authorities. We also see a lack of assessment of regulatory readiness. We also see a lack of regulatory oversight in clinical planning. We also see a lack of oversight of country requirements. And all these factors lead to delays in site activation, delays in recruitment, and eventually delays in timely completion of the study. So how do we overcome these challenges and pitfalls? We have identified various considerations, and these are strategic engagement, and we have also identified considerations for conducting a successful gap analysis. We have identified considerations for supporting clinical operations, for example, labeling and logistics, and we have identified considerations for successful regulatory planning. And in the next set of slides, we'll go into details of these considerations. So for strategic engagement with the authorities, there are various options. For example, scientific advice is often sought from the FDA and EMA, but there should also be considerations for obtaining further advice from other regulators in line with the overall product development strategy. And some examples where this is applicable is in China, Germany, and Japan, where we highly recommend obtaining additional scientific advice from the authorities. And in some countries, instead of scientific advice, we prefer to go through a pre-CTA submission consultation process. And we do that to facilitate site startup. And often in Argentina, Brazil, and South Korea, this strategy works perfectly. So instead of a scientific advice, we can simply ask for a pre-CTA submission consultation. And finally, for genetically modified cell therapies, a pre-CTA submission consultation with the GMO authorities is often required, for example, in Belgium, Norway, and Finland. So these are for strategic engagement. So what do we do for gap analysis? So for a successful gap analysis, we start with the characteristics of the product. So, for example, for gene therapies, we consider whether it is an in vivo therapy or an ex vivo therapy and consider the route of delivery. And for cell therapies, we consider whether it is an autologous therapy or an allogeneic therapy and look at the types of substantial modifications done to the cells. And this is really to allow us to set the parameters for regulatory readiness. And under regulatory readiness, the aim is to review the documents required for submissions in the light of country, local, and regional guidelines and regulations. And a big chunk of this readiness is done on the IMP dossier. For example, we would review the manufacturing methods and starting materials, cells, and tissue testing requirements, and make sure that these fulfill the expectations of regulators in the country of interest. And we also identify any site-specific cell and tissue licenses that are required, for example, for handling, import, export, and manufacturing. And these licenses are required to be included in the CTA submissions in some of the countries. And an additional step that we look at is also documents that are required for RA and EC submissions. And these are specific for cell therapies. And these can be surgery, apheresis manuals. These can be cell therapy manuals and tissue handling manuals. And for genetically modified cell therapies, there are additional documents required for GMO submissions. And some examples of those is the environmental risk assessment, common application form, summary form, and there are also country-specific application forms, such as for Australia, Brazil, Canada, and Japan. And there is also a requirement to have a material safety data sheet that determines the biosafety level for safe handling of these products. So when it comes to regulatory involvement in operational planning, we are involved in IP labeling and logistics. For example, we would ensure that the IP labels comply with local regulations and that the mandatory precautionary language for safe handling and disposal of the product is also included, which is often a requirement in most of the countries. And in addition to the IP labels, we should bear in mind that auxiliary labels are often required for syringes, IV bags that are used for injections and infusions that happen at the clinical site. And these labels should be designed and reviewed by the regulatory team as well. And for logistical aspects of the planning for the trial, the regulators team should also ensure that the country-specific import, export, and transport licenses are also obtained, and these are applicable to countries such as Australia, Brazil, China, Hong Kong, South Korea, Turkey, etc. And similarly, regulatory teams should also be involved in discussions concerning local depot requirements, so that the regulatory team can confirm the licensing that are required and confirm that these facilities do hold these licenses before sites are initiated. And for regulatory planning and submissions, we should bear in mind that the RA and IRB-EC review timelines can be extended to seek expert opinion. So it's really a dynamic timeline that we should consider, and we often look at the shortest and the longest path ever it took for reviews of such when we are planning for the timelines. And we should also bear in mind that there can be additional review bodies at the site level, like institutional biosafety committees or stem cell research committees or advanced therapy advisory committees that needs to often approve the trial before the sites can be initiated. And for genetically modified cell therapies, we often see a requirement for the environmental agencies to approve the trial before sites can be initiated. And this happens in Australia, Brazil, Canada, France, and Spain, just to give you examples. And then there are often requirements to also obtain cell and tissue licenses. And this can be applicable to the clinical site, it can be applicable to the pharmacy, it also applies to the infusion centers that are used. And this is really to license the activities such as procurement, usage, import, and export of cells and tissues handled by these facilities. And we need to make sure to obtain these licenses prior to submissions, as some countries would require these to be included in CTAs. And similarly, manufacturing licenses might also be required depending on what steps are completed at the site level and whether these fall under the definition of manufacturing. And if they do, then the licensing should be obtained for these facilities. And with that, I will hand it over to Elizabeth Shepherd for operational considerations. Thank you.
Thank you, Jan. So layering on to what Jan has already described, I'm going to elaborate on some operational considerations for developing cell therapy. The first being the regulatory requirements for tracking cell and tissue-based products. Shown here are the requirements for the tracking system in the US, but globally, there are equivalent requirements. To summarize these requirements, the system must track the product from the donor to the consignee or vein to vein using a distinct identification code that is traceable throughout the chain of custody and identity and can be tied to all records pertaining to that product. Further, this tracking system and the requirements of the system need to be provided to the consignee in writing before distributing the product. When we consider the logistics for cell therapies, this tracking system is often very complex with many stakeholders. For an autologous cell therapy, the starting material is collected from the same patient that will receive the final product. After collection at the clinical investigative site, the starting material must be transported to the manufacturer with cold chain logistics and/or time constraints due to the product's stability. After manufacturing or expansion, the product requires transport again back to the site for storage and later administration. For an allogeneic cell therapy, there's a product on a shelf for a patient, and there were many logistical steps prior to the availability of that product on the shelf, all of which are part of the tracking system. Once the recipient of the product is identified, there may be a requirement for matching between the patient and that allogeneic product before, again, transporting to the site with cold chain logistics and/or time constraints, again, due to the material stability. For either scenario, these logistics are defined by the patient's condition, the material stability, and the capabilities and processes of each of the stakeholders involved. And all of that must be considered when developing the tracking system. Another important operational consideration is the manufacturing of these cell therapies. This is a newer field of medicinal products, often being pioneered by small biotechs that need to transfer complex manufacturing process to a contract organization. And the time to complete this transfer is often underestimated. Further, the manufacturing of these cell therapies depends on the availability of the starting material, which can be unpredictable, whether autologous or allogeneic. This may require shared manufacturing space or careful consideration around the availability of a product versus study enrollment, for example. It's also important to consider the location of the manufacturing versus location of product collection and administration site, and the feasibility of maintaining the product stability requirements between those locations. In some cases, to ensure that the product remains stable before it reaches a manufacturing facility, there may be a requirement for the collection site to manipulate the starting material. And in this case, it becomes necessary to ensure the site has the appropriate equipment, capabilities, and qualification, or possibly to collaborate with the site to build the appropriate capabilities at the site, which again can impact the study timeline. So bringing all the considerations we've reviewed so far, I'd like to offer a summary of the most common pitfalls we see in this space, all of which can lead to timeline delays and increased costs for the study. The first pitfall is an issue with achieving regulatory requirement due to a lack of early engagement with the regulatory authorities or integration between regulatory and operational planning with regard to product development strategies. The second most common pitfall is delay in site startup activities due to the additional site-level submissions and the licenses and documents that are required for these site-level activities. The third most common pitfall is delays in manufacturing or supply chain readiness due to the complexity of the manufacturing processes or availability of the starting material for the final product. And the fourth most common pitfall is the delay in enrolling the study, because these therapies often target very specific disease subpopulation, often in advanced or rare diseases, where the patient's condition and readiness for treatment must be considered. To exemplify these pitfalls, we'd like to walk through 2 case studies and provide some best practices. In this first case study, let's examine a common hurdle of aligning supply chain and clinical operations readiness. Individually, the supply chain can face hurdles like, again, availability of starting material, transferring and then fine-tuning complex manufacturing processes, or recognizing later in startup or later in that transfer that additional data is needed to support product stability. On the other hand, ensuring a site is ready can present challenges in the form of a limited number of sites that actually have the capacity or the capability for these complex trials, or sites with lengthy startup procedures due to their requirements for additional committee reviews and availability of study documents. To the point of this case study, though, the readiness of each of these entities can be interdependent as well. For example, it may become apparent that additional stability data is needed for documents like a pharmacy manual that a site requires for startup, or development of the manufacturing process or product stability results may reveal a need for sites to manipulate the starting material or handle the final product in a specific way. And this could require additional equipment at the site, additional training, additional feasibility. Overall, though, a lack of readiness in either entity usually leads to a timeline delay. Another aspect of this hurdle is the development of the regulatory required tracking process. As we reviewed earlier, the logistics in these therapies are complex. And so developing the tracking process requires a very thorough understanding of each stakeholder's role in the process and the information they require, as well as the process they're going to use while they take part in the tracking system. If the tracking system for the study isn't developed robustly, this can lead to insufficient documentation or insufficient ability for oversight of the tracking process. It can also lead to a need to add stakeholders and amend the tracking process, or it can lead to an unsuccessful attempt at validating the process that again might lead to amending and revalidating the process. So best practices for overcoming this hurdle of aligning supply chain and clinical operations readiness are to first initiate the development of this tracking process early with involvement from all stakeholders. Build that really robust understanding of each stakeholder's process and the role they play in the tracking process early so that there's sufficient time to address any issues or other hurdles that come up. The second best practice is to consider the IP handling in a clinical setting and include those considerations in the product design and manufacturing plan. This will allow for a very helpful feasibility with the site before bringing them onto the study, and it avoids the issue of running into a hurdle with the site capability or capacity once the study is about to start. The third best practice is to evaluate the site capabilities and clinical flow in the context of the product-specific handling requirement. So again, considering the IP handling in the clinical setting and then evaluating those site capabilities based on the product handling requirements will help to avoid unforeseen hurdles later in the study timeline. In the second case study, let's examine the hurdle of taking a cell therapy study global. When going global, it's critical to consider the product development strategy early to ensure that CMC data is available to meet the country-specific requirements and that submissions can occur on time without need for additional submissions with supplementary data. Without early consideration, it can become difficult or even in some cases unfeasible to include a specific country. Similarly, it's critical to consider the country-specific labeling requirements early so that it doesn't become necessary to develop additional labeling documentation, to relabel IP, or to institute country-specific labels, all of which can lead to a timeline delay. Finally, when going global, it's critical to consider the supply chain logistics. For example, the requirements for importing and exporting human material will vary, as Jan described, as will the transport timeline, which again may or may not be feasible with the product stability. Further, the capabilities of the sites and the vendors will vary globally. So it's important to consider if additional vendors are needed or if we'll need to support the sites in some way for them to be able to execute the study. Best practices for overcoming this hurdle of developing a global strategy for a cell therapy trial is to first identify and confirm feasibility of taking the trial to a particular country by addressing local requirements and regulations for the IP manufacturing and the stability data and labeling of the IP early, and again in the context of the study timeline. For example, if IP will need to be relabeled, are we aligning our timeline for going live in that country with the timeline that it will take to relabel the IP? Second best practice is to assess feasibility of IP supply in the context of site and vendor capabilities prior to the selection of sites and vendors. As we discussed earlier, typically these cell therapies require cold chain logistics for transport and then must be maintained sometimes at vapor phase temperatures on site. It's important to understand, again, for example, if vapor phase temperatures storage is feasible at a site, or if there's a limitation to the amount of storage at the site at vapor phase temperatures. By considering these restraints early, we can potentially mitigate the concern by, for example, providing additional storage to the site. Like many other aspects of clinical research, COVID has added complexities to cell therapy operations. For example, the logistics process and tracking process that we walked through earlier becomes much more complex with flight delay. And so it's important to work with a vendor, whether a logistics vendor or a courier, that can pivot and mitigate these unforeseen flight delays or other transportation impacts of COVID-19. Further, we've seen that site startup timelines can be extended due to the prioritization of COVID-19, or because there's reduced on-site resources at the site. It's important to develop mitigation strategies for these impacts, such as ensuring that all the documents the site is going to need for their committee submissions are readily available well before the submission will be made. It's also important to keep the impact of COVID-19 restraints at the site in mind when planning for the study timeline. Taking this all together, we wanted to leave you with 3 overarching best practices for avoiding common pitfalls in cell and gene therapy trials that ultimately will increase the timeline and the study cost. The first best practice is to include global considerations in the product development strategy. The second is to strategically engage with the regulatory authorities and consider the variation between regulations, again, early and with consideration for the product development strategy. Finally, the third best practice is to ensure communication and alignment between regulatory submissions, clinical operations, and manufacturing and supply chain readiness. More than ever before with these advanced therapies, it's critical to have good teamwork and frequent communication about operational plans to ensure that hurdles can be addressed early and mitigative strategies can be put in place. With that, I think we'll open it up to your questions.
Thank you very much for this presentation. Just a reminder for the audience members, in order to send in questions, you can do so via the Q&A widget. But it looks like we've got some questions coming through already. So the first one in today's session is, what are some recommended mechanisms for validating the cell tracking system and communicating the tracking system to the involved parties?
Sure, I can take that one. So for recommending the recommendations for validating the system, what we want to differentiate is that it's important to validate the system in a couple ways. It's important to validate the system that it's fit for purpose and that it meets the regulatory requirements for the system. In addition to that, it's important to validate the system in an operational way. And what I mean by that is to really do a dry run or a mock run of the system with all the stakeholders to ensure that, first, the system has been clearly communicated, and second, that all of the stakeholders are able to complete their pieces of the process in a way that aligns with their institutional processes or requirements, and also in a way that provides the information needed for the next stakeholder in the process. So again, we recommend conducting a dry run of these processes, which could include the investigative sites as well as the vendors and other stakeholders, or you can do more focused dry runs, for example, with just a manufacturing facility and a courier.
Thank you very much. The next question that we have is the cell tracking system expected to change as the IP progresses through various phases of research?
Yes, I can take that one as well. So the short answer is yes, we would expect that the cell tracking system will change as the research progresses through multiple phases. When we think about early phase trials, we're usually looking at maybe 1 or 2 countries with a couple sites, maybe less than 15. And so the tracking system it can be somewhat more manual or paper-based, and as it likely also will include less overall stakeholders. However, as we progress through the phases of research, it's likely that we're going to expand to additional countries, and that maybe we will have, instead of 1 manufacturing site or 1 depot, we will have multiple depots, and those depots may have different capabilities or may take a different process for example, preparing a cell therapy for shipment to an investigative site. So Once we scale up in terms of global reach and number of sites and vendors, I would expect the tracking process to change and likely become, to a certain degree, more electronic or more automatic, so that these operations can happen throughout the day in various time zones, and with the appropriate oversight compared to a smaller trial that's happening in one geographical region.
Thank you very much. Next question we have is, are there noticeable differences in advice from FDA and EMA?
I can take that one. Yes, indeed. So because the requirements vary between, in the US set by FDA and in Europe set by EMA, there can be different considerations for both continents. For example, cells that are retrieved from the patients might be compatible for one population and not the other one. And one authority might focus on this aspect of the trial, of this aspect of the product versus the other aspects. Or the trial design might be a bit of more focused on, there might be other considerations for EU versus US. So yes, so there can be differences in the advice and what it is actually asking.
Thank you very much. The next question that we have is, what issues have you encountered with the cold chains to and from the manufacturing site? Are there unmet needs that need to be addressed?
Sure. So I think I'll take that question and take it from the perspective of, I think with early planning and detailed communication about expectations and processes, that it's unlikely that you would face issues with cold chain logistics to and from the manufacturing site. Again, kind of going back to operational considerations, and specifically IP handling, I think it's important to early on understand the expectations of the IP handling. So for example, if IP needs to be shipped in a refrigerated state and it must be transported between sites within 24 or 36 hours, we need to be very aware of that as we're setting up the cold chain logistics, and ensuring that the vendor selected for that purpose can meet that 24 to 36 hour window and has multiple options or lanes to be able to meet that 24 to 36 hour window. So again, I think that there's the potential for issues, but with early communication, we can avoid these issues by putting mitigation plans in place and being aligned on what the requirements are.
Thank you very much. The next question that we have is, the auxiliary labels for syringes and IV bags, are these required if you do not supply the syringes and bags, or only if the sponsor provides?
I can take this one. The requirement for auxiliary labels are required to ensure the traceability of the product all the way to the patient, when it arrives to the site and then it reaches the patient. And this is really to ensure also the patient's safety. So irrespective of who provides these materials of syringes and IV bags, there will be a requirement to label all of them to make sure and to trace which patient actually received that product in that bag or in that syringe.
Thank you. The next question is, what are the first steps to consider for developing a cell tracking system?
Sure, I can take that one. So I think the very first step to developing a cell tracking system would be to consider the major handoffs or the chain of custody that will be involved in that cell tracking system. So just like the examples we reviewed, I think first step is to put on paper for an IP specifically, who are the major holders of the IP and the information associated with the IP throughout the cell tracking system. And then layering on top of that, what are the timeline requirements and the disease indication requirement. So like we talked about, many of these therapies treat patients whose condition is somewhat critical. And so, for example, do we need to ensure that IP is on site within a certain amount of time from patient screening? So layering on those timeline requirements to the major handoffs that we understand must happen for product to get to patient. I think that's really the first step. And then from there, it's understanding who are the stakeholders that are participating in those handoffs, what's their process for completing the handoff, and what information do they require, and how are we going to exchange that information between stakeholders. I find it to be a very iterative process to develop these cell tracking systems, and one that requires a very team mentality and a collaborative mentality to ensure we're all aware of each stakeholder's role and what they need to complete that role in the system.
Thank you. The next question we have is, in the presentation you noted common pitfalls in patient recruitment for cell therapy trials. For an autologous cell therapy trial, what are some of the operational considerations?
Sure, I can take that one. So for an autologous cell therapy trial, again, we're talking about IP that is, the starting material is coming from the same patient into which the final material will be infused. And kind of going back to something I said in the last answer, again, it's often the case that there's a very specific timeline that must be followed for between collection of the starting material and infusion of the final product, and sometimes even before collection of the starting material. For example, if the patient is in a somewhat critical state, or we need to ensure the patient is fit for collection of the starting material, we need to take that into consideration when planning out our manufacturing timeline, and ensure that we have good communication with the site who's planning and ultimately going to execute the collection of that starting material. Similarly, after collection of the starting material, it's possible that the patient will need some type of therapeutic support or bridging therapy between collection of starting material and when the final product is available to be administered to the patient. And it's also possible that, based on understanding of the product's mechanism of action, that there would need to be some washout periods between that bridging therapy and the infusion of the end product. And so again, we need to keep that in mind. And of course, putting at the top of the priority list the patient's condition, and ensuring that our manufacturing timeline aligns with what we expect from a patient timeline perspective. So I think kind of a short answer to the question is that the most important operational consideration for an autologous cell therapy trial, especially as it pertains to recruitment and enrollment, is really that cohort management or individual patient timeline management.
Thank you very much. Another question we have is, given the vast array of regulatory considerations, what are the first steps to take for companies when considering their cell therapy product for a clinical trial?
I can take this one. So product development strategy should be set as early as possible, ideally at the time of non-clinical development, to give us an idea of what markets are more suitable to market the product, or to indicate us the goals of the company where they wish to market this product. And once that is set, that gives us a starting point to conduct a country feasibility, and then authorities are contacted to make sure that the design and the CMC aspects of the trial of the product is determined, and then from there we get into clinical feasibility to see where the sites are and if it's feasible to run the trial in those particular sites. And this way, we proactively plan and ensure regulatory compliance throughout every step of clinical development.
Thank you. Another question that we have is, compared to FDA or EMA and perhaps PMDA, how would you describe are the HAs of the rest of the world, e.g. South America, Australia, etc., prepared to have cell therapy clinical trial conducted? Can we expect similarly smooth businesses there as we do in the US and EU?
Yes, I can take this one. So in terms of trial design, obviously there are differences in different continents and countries, and especially as PMDA is mentioned, Japan is particularly concerned about the population. When we talk about cells and tissue therapies, there's a concern that, you know, there will need to be data obtained in Japanese population, and that's the main concern to run a phase 3 trial, for example, in Japan without having any tests conducted using those cells and tissues in Japanese population. And the best strategy in this case is to consult the authorities, and perhaps they would request a bridging study to be conducted before they approve a bigger study to go ahead in Japan. And definitely there are differences, and this should be addressed. However, for Australia and South America, so these countries or continents more follow FDA and EMA advice, so it's not as complicated in those continents to figure out what the requirements are. And certainly a consultation held with these authorities help us run the trial smoothly.
Thank you very much. Another question that we have is, when do you recommend in general the commercial formulation and supply chain to be finalized in the process of the development for cell therapy pipelines?
I can take that one. And so, you know, ideally, the commercial formulation and supply chain are finalized around the time or you know, short after we start to assess feasibility of conducting the research at the investigative site. And the reason I say it's finalized around that time is because we need to be able to have conversations with the sites about how they will be handling the investigational product, the cell therapy. What kind of capabilities are required on their end? Because at that point, they need to make an internal determination about whether they have the capacity to support the research. You know, this area, especially in terms of early research or early phase trials, is burgeoning, and there's a lot of research going on, and these trials are more complex. And so therefore the sites do have some, you know, limitations around their capacity to take on a certain number of trials, or they might need to, for example, ensure a particular type of equipment is available on site before beginning the trials. So in an ideal setting, the commercial formulation and supply chain are final or near final when we're having those early conversations with the site about the feasibility of bringing this trial to their site.
Thank you very much. This is all of the time that we'll have for questions in today's session, but just a reminder for the audience that your questions will be available to be answered at a later date, so you can still send in questions on demand as well. So that just leaves me to thank Jan and Elizabeth for this great presentation, and to Medbase for sponsoring this session. To the attendees, you will receive an email tomorrow telling you how you can access the on-demand version of this webinar, or you can access the on-demand through our website, which is www.business-review-webinars.com. We look forward to sharing further webinars with you, so please do keep an eye on the website just mentioned, and you can also follow us on Twitter at BRWebinars, or join our LinkedIn group, Business Review Webinars, for daily updates. Thank you all once again, and I hope that you have a nice day.